How does your immune system recognize non-self antigens? Imagine your immune system as a vigilant security team constantly scanning for intruders. Its job is to spot anything foreign that could harm your body. But how does it tell the difference between your own cells and those pesky invaders? That's where non-self antigens come into play. These are molecules that don't normally exist in your body and act as signals for the immune system to respond. The process starts in special organs called primary lymphoid organs. Bone marrow is where B cells are made, and the thymus is where T cells develop. During their development, these cells undergo tests to see if they react to your own tissues. If they do, they are eliminated or turned off. This step is called selection and helps prevent your immune system from attacking your own body. Once mature, immune cells travel around your body, always on the lookout. They look for antigens presented on the surface of other cells. These antigens are fragments of proteins called peptides. Special cells called antigen-presenting cells grab bits of pathogens and display them on their surface using molecules called major histocompatibility complex molecules. T cells have receptors called T cell receptors that recognize these peptide major histocompatibility complexes. If the T cell receptor binds strongly to a peptide major histocompatibility complex, it signals that a non-self antigen is present. This triggers the T cell to activate and start an immune response. If the interaction is weak or matches a self-antigen, the T cell remains inactive or is told to ignore it. B cells also play a role. They have receptors called antibodies that can bind directly to antigens. But for full activation, B cells often need help from T cells. If a B cell binds a self-antigen, it usually cannot activate unless a T cell also recognizes that same self-antigen, which is rare because self-reactive T cells are eliminated early on. This two-step process helps prevent your immune system from attacking your own tissues. The entire system works like a finely tuned alarm. When a non-self antigen is detected, it triggers a cascade of signals that lead to the destruction of the invader. The interaction between T cell receptors and peptide major histocompatibility complexes is key. Strong interactions usually mean non-self, and that's when your immune defenses spring into action. Understanding how your immune system recognizes non-self antigens helps us develop better vaccines, improve organ transplants, and find new ways to treat autoimmune diseases. It's a complex dance of cellular signals and molecular recognition that keeps you safe from external threats while respecting your body's own cells.